Microbiology is a subject that deals with the study of microorganisms, bacteria, fungi, algae, viruses, protozoa, a battery of these kind of germs. When I say germ, it does not literally mean that it is a pathogen. Microorganisms can be pathogenic or non pathogenic. But to define microorganisms are ubiquitous in nature. They're found in air, water, earth, soil, etc. The study of microbiology is fascinatingly, wonderfully explorative. Certain chapters in microbiology, studying a course in microbiology, certain chapters include sterilization, which means disinfection to completion, which means complete removal of microorganisms from the surfaces, be it laboratory equipments, be it glasswares like Petri plates, complete removal of microorganisms both in the vegetative state and in the spore state. What is vegetative state? Vegetative state is the active reproductive state, whereas the spore state is a more dormant state. It is a resistant stage of a microorganism, particularly bacteria. So, sterilization is one chapter. Microscopy, using optical instruments devised by the scientist Anton von Leeuwenhoek. The use of microscopes to view microorganisms, the size, the shape, etc. Stains and staining solutions is another chapter after microscopy. That is the use of biological dyes that comprises both oxochromes and chromophores, the ionic component and the color, the chromogenic component, the color giving component, used in staining microorganisms so that it is viewed through the microscope, through a system of lenses, by creating a contrast or a coloring agent that colors the surfaces of bacteria and even fungi. For example, lactophenol cotton blue is used for staining fungus. So, stains and staining solution is another chapter. So, we have spoken about sterilization, we have spoken about microscopy, we have spoken about stains and staining solution, and well, that can be one particular chapter by itself. That is Introduction to Microbiology. Even before understanding an introductory concept on the subject microbiology, we have history of microbiology. The scientists who pioneered and worked their way through explaining what bacteria is, what a germ is, what a pathogen is, what is virulent form, what is avirulent form, etc., what a virus is, etc. Some of the contributors in the history of microbiology is Anton von Leeuwenhoek, who discovered microscopes, Louis Pasteur, who discovered fermentation, Louis Pasteur, who um, discovered vaccine for rabies, viral disease in that a virus that spreads by the bite of a mad dog or a wolf. So we talking about Louis Pasteur, who discovered the vaccine, then Edward Jenner, who coined the word vaccination, vaxa meaning cow, a Latin word. So he understood the concept of vaccination from milkmaids who never developed cowpox virus infection because, or smallpox viral infection, because they were already infected with cowpox virus because they were milking the cows on a regular basis. So we have contribution by Anton von Leeuwenhoek, Louis Pasteur, Edward Jenner. Then we have soil microbiology developed by scientists like Vinogradsky. We have um, Robert Koch, who came out with Koch's postulates that later on explained the concept of medical microbiology. So we have history of microbiology preceding the introductory component or introductory chapters into microbiology, into defining and understanding and broadening one's idea of microorganisms, their removal, their observation through instruments like microscopes, the stains and staining solution used to stain bacteria and fungi, etc., the properties of these stains and staining solutions. Thank you. I've just invited you. I have just got you excited on the subject of microbiology. Welcome aboard to a world of microbes, microorganisms, germs. Thank you. This is Dr. Reena Nelson Antikat from Kerala, India. Thank you.